Here's the plan. I'm going to start off with a Shaggy Dog memoir introduction. I'm going to do some wet lab science. And then, like Jack Gilbert, and perhaps it's the University of Chicago influence where I did my graduate work, I'm going to bring hard science into something softer. I spent most of my career not at Rutgers, but at Tulane University, which is in New Orleans. And while there, I worked on a fungus called Aspergillus, studying a family of very bad toxins called aflatoxins, which are famous for their blue fluorescence and the fact that they cause cancer in very low doses. They are an example of a larger family of compounds called mycotoxins, the bad things that molds make. Huge deal in international food supplies. Most of the time, when we're exposed to mycotoxins, it's because we eat them inadvertently, but we can breathe them in and we can absorb them through our skin. Inasmuch as I have a scientific claim to fame, it's that I brought genetics to the study of aflatoxin before you could clone genes. I did so by clumsily isolating a parasexual cycle, and along the way, I used fluorescence to isolate blocked aflatoxin mutants, and a bit of luck, many of them accumulated colored pigments. I did old-fashioned precursor product biochemistry and painstakingly, over the course of many years, worked out a simple biosynthetic pathway. And then came gene cloning, and with a lot of help from Pern-Quan Chang, Zhu Jiang Yu, colleagues at the US Department of Agriculture, and people working elsewhere, Gary Payne, Dennis Shea, we were able to show that the genes for this pathway were linked in a cluster. In fact, it was the paradigm for secondary metabolic clusters, which I'm proud to say are now some of the easiest things to pick out of genomes. And we did it, penicillin's one too, and hey, we're just a small group of ag workers. Now, as I said, mycotoxins are largely funded because they cause trouble in food. We know a lot about how they enter the diet. But in the last, say, 20, 25 years, people have gotten worried that they may also be contributing to a phenomenon called sick building syndrome. Now, that's a very moldy house. But very often when people suffer from supposed sick building syndrome, you can't see the mold. The term itself is not widely accepted. Syndrome means running together. So you have a lot of very nonspecific symptoms. And they're so nonspecific that almost everyone has experienced at least some of them some of the time. Sore throat, dizziness, insomnia, perhaps depression. The idea being that when you leave the building, you start to feel better. Back when I had a son at the University of Chicago, paying a lot of expensive tuition bills, I did some consulting, something I don't like to do. And I used to represent insurance companies and say, I think it's almost impossible to breathe enough mycotoxin to get sick building syndrome. And I still believe that. You really have to inhale a lot of spores. And not only that, the symptoms of sick building syndrome are much broader than anything that you get from any one mycotoxin. Okay, I have to set this up. So I was very active in the early days of fungal genomics, politically, trying to bring money to sequence fungi because they were so cool. And in August 2005, just 10 years ago, I was about to go and spend a sabbatical from Tulane at the, what was then called Tiger, now the Venter Institute, because my fungus had had its genome sequenced. Never got there. And so from now on, I'm going to tell you what happened. On August 28th, 2009, a great big hurricane hit New Orleans. My husband and I evacuated the day before. Now, I want to point out something about New Orleans. It's at the mouth of the Mississippi River. It is just south of a huge inland lake called Lake Pontchartrain. The Mississippi River winds through it. There's swamp all around. And this particular elevation map, everything in blue is below sea level. So a lot of the levees failed at Katrina. And this engineering diagram basically tells you what happened. This huge inland lake, Lake Pontchartrain, just came pouring into the city of New Orleans. This is how it looked from the air. Here's a view from the ground. And this cartoon appeared in the New Yorker just a few months later, and I don't know if you can read the caption, but it says, "When the waters subside, the problem's going to be mold." Let me show you a couple of pictures. Zhu Jiangyu, who helped with the aflatoxin paradigm, had a ranch-type house where he got 12 feet of water. He took pictures. Pern Quan Chang also flooded, but he didn't take pictures. Now, as I told you, I had left the city. We didn't get back. We went to New Jersey, and I knew what my house was going to look like, so I called a buddy at Rutgers and said, "Hey, can I come to your lab and make some media?" It was sort of sublimation. I've got to handle what's happening with my home. And so the first time I went home, I took pictures. I made swabs. And that's what's gotten me interested in what I do now. Here are some of the pictures. It was pretty bad. It was emotionally bad. You don't like to see your house alive with fungi. And remember, this is the organism, life form I've studied all my life, eating my house. And I mean, we basically lost all of our furniture and possessions on the first floor. Now, what the pictures can't tell you is how it smelled. I have never smelled anything like that. And I'm sampling my house, and I start to feel sick. And I'd go and sit outside, and I'd come in, and I had a conversion experience. And I said, maybe there is something to sick building syndrome. And maybe it has nothing to do with mycotoxins. Maybe it has to do with these gas phase molecules. So I'm not the only one who thought that, but remember, I'd done all that consulting. I knew a lot about sick building syndrome. You know, Pasteur says chance favors the prepared mind. Well, so does disaster. Scandinavians have hypothesized that volatiles might be involved. So meanwhile, I went back to Rutgers to culture my molds. They knew I was on sabbatical. They invited me to finish sabbatical there. And then they offered me a job, and they hired me as an associate vice president to start an office for women in science. Meantime, in my spare time, I ran a lab. So let me tell you a little bit about volatiles, volatile organics. They're these small compounds that easily evaporate, and we all know a lot about them. Like some of the most famous are solvents we use in the lab. Other famous ones are anesthetics, which we've been using a long time to kill pain. I hadn't really given it any thought that everything we can smell is volatile. And I really hadn't ever given that any thought. So once you get interested in volatiles, you get interested in odors. And hey, fungi are famous for some of their smells. Truffles are thought to be aphrodisiacs when you inhale them. And then there's the stinkhorn that only flies can love, one of the most nasty smelling life forms on the planet Earth. So anyway, I had this little lab on the side, and we set out to say, well, what fungi did I have in my house? What VOCs did they make? And then something I've done that I don't think anyone else has done is I've used genetic models to interrogate them. We didn't find any new fungi. We spent a lot of time on this. Trichoderma was the main genus. And we devised these little microhabitats where we grew the fungi on sheetrock, and we collected them, and we did GC mass spec, and we found what lots of other people make. Fungi make lots of volatiles. Depending on how long you grow them and what temperature and what species, you're going to get a different profile. And we didn't find anything new there. But remember, I said that this is something I don't think anyone else has done. We brought our volatiles to genetic models. And I started with C. elegans. You know, you can grow it in Petri dishes, and I thought it was going to be best. But hey, basically, it crawls away from the fungal volatiles and gets away. So we had to abandon it. You can't even find the dead bodies. With the remaining models, we are spread kind of thin, but they all have their own advantages. We've done two things. We either expose the model to the growing fungus so they get all the volatiles, or we go and we buy individual volatiles, and then we can control them. And when we bought these individual volatiles, we bought the cheapest ones we could find. Richard Hung, who'd been an undergraduate with me at Tulane, was my first graduate student. He pioneered the Arabidopsis. I'm not going to spend much time on this, but I want to say that we're constantly having to reinvent protocols. Exposure systems are very tricky. You have to keep things enclosed. You have to keep them on a shaker. And this is hard work to do. We have had a great deal of success, however, and here's an example of one of our volatiles, a compound I'm going to be talking a lot more about, 1-octanthreol. It has a common name, mushroom. It's why mushrooms smell the way it does. It inhibits seed germination in Arabidopsis, and if you get the concentration up, it kills the plant. We've also found volatiles that make plants grow better, but I'm not going to go there right now. Meanwhile, I'd hired a young neurobiologist named Archie Inamdar to help me with the C. elegans, and she said, this is a rotten system, Joan, let's move into flies. So again, inventing exposure systems. Sometimes we expose the larvae to growing fungi. Sometimes we put the chemical standards into the adult flies. Here are some data with growing fungi, and trichoderma actually was fairly toxic. And one of the wonderful things about the fly system is you can see all kinds of effects of metamorphosis along the way, and we've done many different compounds. Each one is giving us a different set of results. But back to 1-octanthreol. As we were testing individual volatiles on adult flies, we discovered that 1-octanthreol and other 8-carbon volatiles was really very toxic. If you were to do this with toluene or benzene, most of the flies would still be alive after 8 days. And with 1-octanthreol, they're all dead. And not only that, they move funny. And Archie, who knew something about using flies as a behavioral test, recognized that you could run them through a behavioral test that's been developed for Parkinson's disease. And these flies were acting like paraquat and other known agents that cause Parkinson's disease. One of the great things about flies, I'm a geneticist, is that, they're a model. So you can, a lot's known about it. And this is where the tyrosine hydroxylase genes are expressed in the Drosophila brain. And so you can send like $20 to the Indiana Stock Center and buy yourself flies that have GFP hooked right up in front of that gene. And we did that, and we could see that you had downregulation. And we've published a number of papers in this field, just to give you a little bit of data. We also showed that there were effects on metabolic products of dopamine when you test with antigen reactions, and that this led us to believe that there was something going on with dopamine handling. There's a group at Emory that has some human cell culture cells, embryonic cells that have lines in which these genes are underexpressed, and we were able to show that when you expose with the vapor level dopamine, you get very much less uptake. And then we brought it back to flies, and we overexpressed this VMAT gene, and we were able to cure our effect. And as I said, not going to go into detail there because I want to talk a little bit more about this one volatile that we've gotten interested in. We had been working on it by about a year before I had this, like, why are they selling it so cheaply? I mean, it's really a hot compound. Well, it's, as I said, a compound with a common name. It has 2 enantiomers. One smells real mushroomy. They both smell kind of mushroomy, but the other one is more moldy. It's a breakdown product of linoleic acid. I don't think you can see the pathway there. It is manufactured on a large scale chemically to make cheap mushroom soup taste more mushroomy, pizzas, things like that. But this is the really interesting one. It's the compound found in our perspiration that attracts like the malaria mosquito and other mosquitoes. And you know how some people attract mosquitoes and others don't? Almost certainly it's the fungal microbiome component of their bodies that makes some people more susceptible. It also deters some insects. I had never heard this word, semiochemical. I'm kind of weak on chemical ecology. I've learned a lot. 1-octanthreol is a semiochemical. This is a word big in entomology. Most of us know about pheromones. That's when semiochemicals are intraspecific. There's a whole vocabulary of semiochemicals. And many of them are volatile. Many of them are in the gas phase. They signal for insects, things that are in the terrestrial environment. In a lot of ways, 1-octanthreol is a poster child because it is very biologically potent and it functions in many ways. But remember, I'm a geneticist, I'm reductionist, and I said, hey, there's this huge descriptive literature in entomology about what 1-octanthreol does, and I found that it's really toxic in several models, but what's it doing? And Nilgen Tumer in my department had just acquired the yeast knockout library with a robot, and a young man named David Poo just wanted to do a master's degree. He was very certain on what he wanted to do, and he wanted to study yeast. So I said, okay, let's see if we can run 1-octanthreols for the yeast knockout library. We had to get the concentration way up to kill the yeast, but we did. And it was a very clean study. He found 91 of the knockout strains were resistant. We classified them into very resistant, moderately resistant, less resistant. And then we're able to categorize them into functions, and many of the knockout genes were involved in endosomal transport and in targeting. And let me tell you, this research keeps increasing my vocabulary. I had never heard of the retromer. The 5 most resistant mutants all mapped to the retromer. Here's a little interaction map that we built about it. And remember, this is a little bit backwards. These are the genes that when they're not there, 1-octanthreol is no longer toxic. And so we haven't published this work yet, but we find it very attractive that most of the studies done on the retromer have been done by mammalian cell biologists who find that they're very involved in neurological diseases. It's provocative. Okay, with that background, I'm now going to switch into the softer science part of my talk and answer the question. Remember, this is a genomics meeting about the volatile. I'm an all fungi, all the time kind of person, so I missed this word if it was around until last year when 2 people who worked on fungi used it in their title, one on Aspergillus fumigatus, an important fungal pathogen, and the other on the dynamics of kefir, which is a milk fermentation that has a lot of yeast in it. Neither paper gave a definition, they just used the word. So what is it? Now we have to get into etymology and follow out where it comes from. Well, as we all know, since we're in this field, genome is a word that was coined in the German language by a botanist, and it entered English around 1930. And it came in turn from an earlier term, chromosome, which is a good hybrid word that had to do with colored body. And it's interesting, when you go into this etymology, you find that when Homer used that Greek word for body, he always used it as the word corpse, for whatever that's worth. In linguistics, this kind of word is called a portmanteau word. And it comes from, God bless him, Lewis Carroll, who used it in Through the Looking Glass. Perhaps you know Jabberwocky, all Mimsy were the borough groves and the slides mows out grave. And at that time, when Lewis Carroll was writing, there was a kind of suitcase that had 2 compartments called a portmanteau from the French. And here he is describing to Alice about his words. You see, it's like a portmanteau. There are 2 meanings packed up into one word. In contemporary English, smog and brunch are very good examples of portmanteaus. So our word genome is sort of that way. And as we all know, it's part of our story that the genome had been around for a long time, and Roderick took the word and applied it to the name of the journal Genomics in the early days of the Human Genome Project. Another way that this kind of word can be described is as a neologism. It's a newly coined term. It's a newly coined term that maybe has not yet made it into the language. And when it finally makes it in, then it's no longer that, it's a mainstream word. So here are some examples of ones that have made it mainstream. Nymphette, cyberspace, selfie, Xerox. We in the omics field are coining them right and left. And it's sort of an industry. And I found this little cartoon that I thought was cute. Genomics, transcriptomics, proteomics, metabolomics, and then it has comics, doesn't quite rhyme, in the same picture. I've added economics, Reaganomics, Abenomics, Oxford English Dictionary. The OED dates to Victorian England, which may have been the last time when people thought they could know it all. And we guys are sort of onto this again. They're going to catalog all the words in the English language. And I loved the OED. I regularly turn to it. And they have pointed out that this -ome suffix actually is used in 3 ways. And that the way it's used in our profession is this notion of all constituents collectively. And it's a false etymology. It has nothing to do with that Greek and the chromosome and the body. It's a false etymology that we all think we know what it means. In the early days of genomics, when I used to try to raise money for fungal genomics, I used to say that the real Greek root was from a term that meant give me a lot of grant money. So it's spreading. We have the genome, the phenome, the otherome, the unknown. I love the unknown. Some people like the omics, and they point out, hey, you do this, you're a new paradigm guy. Jonathan Eisen has a site called Bad Omics, and he nominates the worst examples. He says it's a language parasite. This graph, I've been told there's somewhere between 800 and 900 neologisms in which we have ome added on. People really love doing this to make themselves feel important. And as I was reading for this, I came across this citation. Why is it spreading so much? Somebody has thought maybe it's because it's associated with that thing that if you meditate, and maybe we all just think that this is great. But be a little bit serious. Words carry connotations, and all of the new omes are getting further and further from the central dogma of the genome, and maybe the transcriptome and the proteome. And when you use a word like volatile, at least to me, it implies that you're close to the gene. And here's a paper published also last year, another fungus, yeast, in which somebody knocked out a gene that's involved in producing some aroma compounds, some volatiles, and found that it attracted fewer flies. The wild type, lots of flies. The knocked out, not so many flies. Now that's a nice genome, maybe volatile. But most of the time when fungi are making volatiles, it's not necessarily quite so close. If you know anything about fungi, you know that they have their stomachs on the outside. Their nutritional mode of life is they pump all these enzymes and acids and effectors and who knows what out into the environment. And depending on what they hit, they can get all kinds of things resulting. And the profile of volatile organics is going to be very dependent on the substrate. Many of them are not direct metabolic products. I mean, lignin degradation is a good example. You've got all these peroxides going out there. Suppose you're hitting some man-made synthetic chemicals. I used to think about that in New Orleans. All the stuff under everyone's sink was part of that mess that was getting eaten up. So just a bit of warning. How far from the genome can you be and still have an ome and have it mean anything? Ome is a nice, convenient shorthand word, but we should also recognize that the words carry connotations, and at least for fungi, volatile may not always be carrying the right connotation. And also to say that we're not the first people to study aromatics. Some of the best chemistry, I've loved studying volatiles because it gives me an excuse to read, have been done by perfumers, and they talk about aromas. And they have all kinds of ways of collecting them, and they don't need to talk about a volatile because they know more than most of us people do. Or even more, the people who study wine. And they talk about terroir and the soil and the way it's handled and the culture and everything. Most of the terroir has to do with the volatile profile. So here's another cartoon I found. It shows the Institute of Neologisms, and we are one of the big biology producers, us in the omics community, we in the omics community. Some of them will make it, some of them won't. I gave you an example that for my own education in genetics, we used to have a lot about the fine structure of the gene done in mutational analysis, and Seymour Benzer talked about cistron, recons, and mutons. Cistrons survive. I haven't heard anybody talk about a recon since I was in graduate school. So here's another quote from Lewis Carroll. "I see nobody on the road," said Alice. "I only wish I had such eyes," the king remarked in a fretful tone, "to be able to see nobody, and at that distance too. Why, it's much as I can do to see real people by this light." Our omics neologisms may lull us into thinking that we know about things better than we do. And if there's any lesson I've learned from genomics, having been an observer since the early days, is that everything we find out, it's a lot more complicated than we thought at the beginning. And here's a sobering thought from the world of volatiles. We've been using anesthetic gases for over 150 years. We know what they do, but we don't know how they do it. Although, among the things they do is put you to sleep. 3 forms of anesthesia. We've all been sitting for quite a while, so I want to end by thanking members of my lab, especially Jason Richardson, who's helped me with the Parkinson's work, the National Science Foundation, and Rutgers. And here is the mycologist version of that poster about when life gives you lemons, make lemonade. I say, when life gives you mold, make new research. Thank you for listening.
All right, thanks so much. I think we have time for a few questions before the break. All right, we've already got one over here. Great.
I have a question about the mold itself have a self protection system for the volatile they make, and also is there a designated organelle to make those organic volatile? Thank you.
Yeah, the volatiles that occur, and I like to call them volatile profiles, not volatomes, are a real mixture of things. Some are biosynthetic pathways. Some of them are these random breakdown products of the substrate. And you can't generalize. I mean, you will notice, unlike most of the talks here where you're talking about huge things, I spent most of my time talking about one compound. And it turns out it also has effects on fungi, and it inhibits spore germination. If you get the concentration up, you can kill fungi with it. I could probably spend the rest of my life, and probably will, studying that one compound. I'm putting a lot of weight on 1-octanthreol, but there are terpenes, there are aldehydes. There's a whole world of these odorants, and I suspect that many of them have very profound biological effects. I often end my lectures by telling people, stop ignoring the headspace. Or the next time you open an incubator and smell that smell, don't ignore it. They're talking to each other. And we really haven't developed the tools for doing a lot of the volatile chemistry. It's a whole new horizon.
Very nice. Streptomyces.
Aha, geosmin.
Well, I'm a soil scientist, and of course, from the beginning, and I guess when you smell soils, and you smell streptomyces on a plate, they're very similar.
Yes.
What kind of just feedback and what types of volatiles might you expect in something like streptomyces to give that very unique, consistent odor, which may be a little bit more independent of substrate, or maybe there is a complex relationship
Yeah.
between that?
Well, a lot of the streptomycin odor is geosmin, and the name was given, I think, back in the 50s because it smells like soil, geo, earth, whatever. It comes from a terpenoid kind of pathway. Fungi make it too, but streps make it better. And interestingly, it's a compound that we humans can detect in incredibly low quantities, so it's hard to isolate. You think you have a lot of it, and it's just because we can sense it. There have been some studies done in Germany by a Swede named Stensmeyer that shows that it is very toxic to flies, and he and I have talked about it. I'm going to do 1-octanthreol, you're going to take geosmin because we're both working in fly systems. We've tested it in Arabidopsis, and it doesn't do anything. We were kind of disappointed. And because it's so expensive, an entomologist gave me his supply just as he was retiring. We haven't been able to get it up to a level enough to do anything in a yeast knockout library. But I find it interesting that evolutionarily, we humans can detect such incredibly low quantities of some of these things. And 1-octanthreol is in that category. I don't think at the low levels when it smells like mushrooms, it's at all toxic, but the dose makes the poison. And when you have an enclosed space where the mold odor gets high, I think it is getting into the level where it may be affecting our neurological system.